Hi guys, today we're starting a new unit which is all about DNA and how it works. So we're just going to get right to it. This page is in your OneNote. It's under a new unit called Unit 4. All right, so first of all, you might already know this, but DNA is actually sort of an acronym. It stands for the really long name of DNA, which is deoxyribonucleic, which is all one word. And then the second word is acid. Um, and really what this means is it just tells you the different parts that make up DNA, basically. Deoxy means that it doesn't have oxygen. Ribo is because the sugar is ribose that makes up DNA. And then nucleic acid is because you think back to biomolecules, it's a nucleic acid. So that's how it gets its name. Just one interesting thing about DNA is that it's still like a pretty recent discovery in terms of like the history of the world. The structure of DNA was discovered in the 50s, the 1950s. So it hasn't even been 100 years yet since people even started to understand what DNA is and how it works. And there's still tons of discoveries happening all the time about human genes, what they do, what they code for, how they can cause diseases, and all that sort of stuff. So it's a really interesting science. A lot of times it falls under genetics or genomics, but we'll talk more about it as we get into some more stuff later on this fall and spring. But just so you know, it's like not a, it hasn't been around for a super long time, the knowledge of DNA. So, okay, here's a picture that looks like DNA. Hopefully, you've seen it before, maybe, but it's a twisty ladder shape. There's two main parts that make up this twisty ladder structure, which are called the backbone. And that's sort of the outsides of the ladder on both sides. So the bottom would also be the backbone. And then the rungs of the ladder that make it up are nitrogenous bases. And a lot of times you'll hear me just call them bases and leave off the nitrogenous. Nitrogenous just means that they have nitrogen. That's the only reason it's called that. I want to go over here really quickly and just cover this. So the shape of DNA is called a double helix, which means there's two strands that are twisted together. Helix means spirally. And then DNA can organize into chromosomes, which we'll talk about more in the spring, but you might have heard the word chromosome before, and chromosomes are just made up of DNA. It's a way that your body can organize your DNA because otherwise it would be just so much and really hard to move it into new cells, which your body has to do when it has to grow or heal a cut or anything like that. And so it organizes the DNA into chromosomes, which humans have 46 chromosomes total. And they're actually in pairs because you get one from your mom, one from your dad, half your DNA from your mom, half your DNA from your dad. And so I'll just say that it's 23 pairs. So you have two chromosome number ones, two chromosome number twos, all the way up until 23. The 23rd pair is the pair that determines whether you are a boy or a girl. If you're a boy, you have XY chromosome, and if you're a girl, you have XX chromosomes, two X's. We'll talk more about that next week. Okay, let's get back to it. So if we come back over here, one of the most important things that I can tell you about DNA is how it codes for your traits. And how it codes for your traits is by the order of the nitrogenous bases. I'm going to say the order of the bases determines your traits. And that's what literally like my bases being in a different order is the reason that I look different than you. And the reason that you look different from everyone that you know, unless you're an identical twin. There's four bases that make up your DNA. They are called adenine, which we call A, thymine, which we call T, cytosine, which we call C, and guanine, which we call G. And from now on, you can always just refer to them as A, T, C, and G. You don't have to know their full names, really. It's good to know them, but you can always just refer to them as A, T, C, and G. So if you look at this like DNA strand, it's really more like each half is a different order of letters. So we'll talk about this in a few minutes, but if this is A, T, C, G, there's actually another half that's pairing with that, and that is T, A, G, C. And I'll talk about that more. But if this was different, this A, T, C, G, if it was A, T, 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 that person would have a different trait than the person who had A, T, C, which is amazing that those tiny little changes actually make up you and make you look the way that you look, and in a lot of ways be the way that you are. Okay, so now let's zoom into this a little more. So if I come down here, the monomers of DNA are called nucleotides, which hopefully you remember from the biomolecules unit. So this picture here is actually showing two different nucleotides. The first one is they're facing each other. So I'll do green. This is one nucleotide right here, and this is a second nucleotide. There's two of them. And I'll label, zoom out a little bit and label this as one nucleotide. And this is also one nucleotide. Within the nucleotides, there are three different parts, and you guys are going to be building this, or maybe already have built this, depending when you're watching this video, in class this week. So I'm going to zoom into just this one nucleotide over here. The circle is a phosphate, which probably looks similar to ATP, and that's intentional because ATP also had three phosphates. Nucleotides have one phosphate. And then you also have, I'm going to do different colors for each. This is a sugar, this pentagon. I'm going to make it. It's a 5-carbon sugar, and that's why it's a pentagon, because pentagons have 5 sides. The name of the sugar is deoxyribose. And you know it's a sugar because it ends with ose. And that is a major reason that DNA is named what it's named. And then finally, the last part of this is the actual base. So this is, I'm just going to write base. Oh, that's the same color as the other one, so I'm going to change it. That's the base. Now, the other nucleotide is a mirror image. So this is a base. This is deoxyribose, which is a sugar. And then finally, the phosphate. Nucleotides are held together by covalent bonds, which is what this arrow here is showing. So this is a covalent bond. Remember, those are super strong bonds. This one would also be a covalent bond that's holding together the phosphate and the sugar. And then down below, the dotted line that's connecting the two bases in the middle, that's a hydrogen bond. So remember that they're a little bit weaker. And that's because in a few minutes, when we talk about replication, we're going to actually cut that those two nucleotides in half so that we can double the amount of DNA that we have. Then one other thing I just want to say is that the phosphate and the sugar together make up the backbone of the DNA. So phosphate plus sugar, this section here, this will make up the backbone of the double helix. And that's on both sides. So over here, this is the backbone of one side, and then the sugar and the phosphate on this side are going to make up the backbone as well. These bases are the rungs of the ladder going up and then twisting. Okay. What's nice about this is that certain bases only pair with each other every time. So there are certain base pairing rules that I'm going to tell you now. And they are that A always matches with T and C always matches with G. And a tricky way or easy way to remember this is at cardinal gibbons, which is where you are. So just an easy way to put that together. AT and then C and G. Always pair together. So now let's talk about how DNA makes copies of itself, because that is really one of the most important things about it. Because remember from cells that you started out as one cell, and then that one cell copied its DNA and replicated a bunch of times to make the trillions of cells that you are now. And every single one of those cells has the exact same DNA genetic code that is you. So to do that, this process called replication has to happen. So I'm going to say replication happens so that the body can make exact copies of cells. And that's important for growing and healing specifically. Those are the main ways that it's really important. We're going to talk more about how the cell actually copies in the spring, but for now, we're just going to talk about how the DNA copies. So here I have placed for you an image of DNA. What's happening here is that the DNA helix is unzipping, which it tells you right here in this picture, that the two strands are unzipping. And in this picture, it doesn't show the bases, it's only showing the backbone. So it's only showing the sugars and the phosphates. DNA is crazy because it only can copy in one direction. And the strands are, it's called anti-parallel, which means one strand runs forward and the other strand runs backward or upside down. So it's like this with your hands, that one's going up and one's going down, and in the middle, the bases are pairing with each other. So I'm gonna actually add that. I didn't have it on here, but I'm gonna add it. I'm going to go over here to the side. This is just a helpful little video that I put here in case you want to watch it. You're welcome to watch it. It's optional. Okay, so I'm going to go over here and I'm going to say anti-parallel means that one strand of DNA runs forward, the other runs backward. And then I'm going to go back over to the other side to show you that a little more specifically. So here I go. These numbers that are the 3 with the little apostrophe and the 5 with the little apostrophe, they're showing you the direction that the DNA is going. So you can see here that the 3 apostrophe, which we call it 3 prime, is on the end and the top strand, and the 5 prime is on the end in the bottom strand. The 3 prime is running in the right direction. That's easy to copy the DNA. So what happens is it's copied continuously. And that's why this blue line is just a nice little, like, clean, straight line because the DNA is just bam, 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 copying the whole way down. The other one is backwards, and the enzymes don't work backwards. So they have to work forward, which means they start like back here at the beginning. And then they work towards the end. So this one has to be copied in little fragments. And those fragments have a cool name after a scientist, which is an Okazaki fragment. At the end of replication, what you get is two exactly perfectly identical strands because on the top, let's say you had, I'm just going to do an example. Let's say that on the top you had A, T, T, C. That means originally it was paired to the other light blue line. So remember, A goes with T, so this would have been T, this would have been A, this would have been A, this would have been G. Now, as the DNA is copied, the new strand is complementary or matching to the old strand. So that means that this will be T, because A goes with T, this will be A, this will be A, and this will be G. On the bottom, this will be A, this will be T, this will be T, and this will be C. So now what you have is two exactly matching strands of DNA. One will go into one new cell, one will go into the other new cell. And that is amazing. So because each new strand of DNA will have half of the old letters and half of the new letters, we call that semi-conservative. So if I'm going to put that into words, what that means is that one half of new double helix is saved or conserved from the old or original DNA, and the other half is newly paired. Now, if you're thinking like, where do these newly paired letters come from? Like, where did I get them? You got them from your food that you ate because you ate plants or animals that had DNA that your body broke down. And then those free DNA pieces can be used to make new DNA of yours by changing the order of the letters, which is amazing. So then you have a semi-conservative situation. The top strand, the one that's copied in order, that's called the leading strand, which I kind of wrote over the top, so I'll just write over leading. And the one that's copied backwards in Okazaki fragments is called lagging because it takes longer, so it lags. In order to do this, your body and your cells have to use specific enzymes, which is why we talked about enzymes before we talked about this. So you'll notice they all end in ACE because they're all enzymes. And their names also match to what they do. So the first one is polymerase. Polymerase's job is to make polymers, which means it sticks things together. So the job of polymerase is going to be adding new nucleotides to growing DNA strand. So the polymerase's job is to match up the right bases one by one by one. If it matches up the wrong bases by accident, that's called a mutation. And we'll talk about that, not in this video, but we will talk about it. The second one is helicase. Helicase sounds like helix, and that's because its job is to unzip the helix. It's like a zipper. Some of you guys did that one for your cereal box. So helicase unzips the hydrogen bonds of old DNA. I'm just going to leave it at that, of old DNA. So then it splits it in half. I'll say like a zipper. Ligase, its job is only on the lagging strand. It has to glue together the Okazaki fragments because if you look back at this picture, there's gaps between them. So they have to actually glue them together. So ligase glues together Okazaki fragments on lagging strand. And then the last one is primase. And primase's job is to add little primers, is what they're called. They're little tiny segments of code, which is what we call DNA. The A's, T's, C's, and G's, the order of them is the code. So primase does that so that polymerase knows where to actually start replicating. So primase, I'm going to say adds primers, which are small nucleotide segments to trigger start of replication. And those are the enzymes that are responsible for this. There are a few others that have smaller jobs or more complex jobs that we're not going to talk about. These are the main ones. All right, finally, we're going to practice replicating. So what you will see is a segment of DNA code. Your job is to try to replicate it. So find the complementary base pair. Pause this video, try to do it. When you're done, unpause me. Okay, great, good job. So the answer that you should have got, hopefully you got this, is T goes with A, then A, same thing other way, C and G, C and G, G and C, A and T, T and A. And then you have created a new strand of DNA, replicate it. And that is it for this video. So great job, and I'll see you soon. Bye.